Okay, COVID-19, is there a link with autism spectrum disorders? Now, I'm going to try and cover quite a lot of material, really as a context for the fact that we don't quite know the answer to the title yet, but I'll tell you where we've got to. But because I'm going to touch on a large amount of material, I've provided quite a detailed reference list, which will be available through the conference website for anybody wanting to follow up on things I mentioned. Right, so the issues I'll try to cover today are: what is COVID-19? Is it linked to other factors like global warming and population growth? How does this pandemic compare to previous ones? How does it compare to previous coronavirus outbreaks? Are COVID-19 risks evolving? What do we know about individual susceptibility? Sorry, I'll just go back to that one. What do we know about spread of the virus? Are people with autism or ASD more susceptible? And what can be done? Now, to address these questions, we need to touch on a range of other factors: the global context and the rise of novel viruses, the nature of the virus, how it spreads to humans and why it spread around the world so fast, how it gets into the human body, the systems it affects, how we can protect against it and limit its effects, and how we should prepare for similar events in the future. Now, when you go to the literature, and if people do go to the reference list and follow things up, you'll find an awful lot of terms which you might not be familiar with. So just to mention a couple of the ones that you may come across and wonder what they mean. COVID stress syndrome is a recent term that's used for an interrelated group of features that people often present with who are affected by COVID-19. Compulsive checking and reassurance seeking, COVID-related fear of danger and contamination, fears of COVID-19 related socioeconomic consequences, COVID-19 related traumatic stress symptoms, and COVID-related xenophobia. Now, that's a group of features that are commonly associated with each other in people who are having difficulty coming to terms with having had COVID and recovering from it. Coronaphobia is a specific reaction to issues related to COVID-19, like other people coughing, related media coverage, food restrictions in local supermarkets or shops, tension and anxiety, which would be consistent with a specific phobia, together with those other factors. And there are a number of papers suggesting that that's a discrete condition now. Studies of risk factors for anxiety in those affected have identified what's now called cyberchondria pandemic and trait health anxiety for coronavirus, or coronavirus anxiety. In the British media, we get a lot of press releases talking about R number or reproduction number. Now, that's a term that's used as a broad figure for whether the rate of COVID in the population is dropping. And 1 is where the level in the population is static. Levels below 1 suggest that it's receding and the numbers are dropping, or numbers of 1 and above 1 suggest that the numbers of cases in the community are growing. Now, we'll have to put this in a broader context. Why is it that we're starting to see more unusual viral outbreaks than was ever the case previously? And they seem to be presenting with increasing frequency. Now, a British scientist called James Lovelock coined the term Gaia to denote the Earth and its biosphere as a complex self-regulating system. He described how our destruction of the ozone layer is resulting in global warming, the shrinking of the glaciers and ice caps, and is causing massive climate change. For Lovelock, there are various systems that interact with each other, and they can largely be put under economic systems and how those are affected by the physical environment and by the social environment. The physical environment, things like the effects of receding ice fields, rising water levels, greater desertification, etc. And the social systems and the effects of things like mass migration because of changes in climate and other circumstances. Global warming is causing changes to our world, melting the ice caps, warming the continents and seas, and causing desertification. Changes to local ecosystems are bringing many diseases into more regular contact with man. SARS, MERS, Ebola, Zika virus, and most recently SARS-CoV-2 are only a few from a large and growing list of these types of disease, which are a result of man impinging more and more on what was previously wild parts of the world. COVID-19 and its effects on our attempts to address climate change and global warming. The United Nations Department of Economic and Social Affairs has a detailed 17-point plan to address a range of global issues. These include famine, access to clean water and sanitation, limiting pollution, improving clean manufacturing, reducing global warming, addressing poverty, improving education, gender inequality, and access to energy. And 13 of its action points have been hampered by the COVID pandemic. Now, I'm not going to dwell on these slides because people can look at them at their leisure. I'll put the presentation on the website. COVID-19 is a disease caused by one of a group of viruses known as beta coronaviruses. Most coronaviruses are found in other species, but not in humans. COVID-19 is an example of what's called a zoonotic virus. It can spill over from other species to humans and vice versa. SARS-CoV-2 is the virus that causes COVID-19. COVID-19 is the term for the symptoms that people who are affected by the virus show. It's the third recent coronavirus to cause an epidemic. The earlier ones were SARS and MERS. All three are beta coronaviruses, and when contracted, all three can cause severe disease in humans and some can cause death. Now, coronavirus is tiny. A single coronavirus is roughly 500 times smaller than the width of a fine human hair. Put another way, it's 10 times smaller than a droplet expelled on human breath. You can see it's the third bottom of the things in this sequence, going from a fine grain of sand down the way to Zika virus at the bottom. So coronavirus is roughly 0.1 to 0.5 of a micrometer. So in size, this, well, I always think it looks like a hand-knitted thing by a grandmother. The diameter is roughly 100 nanometers, and it's made up of a number of components. The one that's been of most interest is the spike trimer, the little red pieces on the top of the ball on the right-hand side of the picture. Now, the spike trimer is the protein spike that latches the virus onto cells and allows it to infect them. That's been the focus of the majority of the vaccines that have been developed, and the spike latches onto ACE2 receptors on the surface of cells in the human body, and that's what enables it to gain entry to the cell. Now, the timescale for that sort of invasion of you as a person, if you contract the virus, is it takes about 10 minutes for the virus to enter a cell. It takes about 10 hours for that virus on entering the cell to make intracellular viral copies, and the number of copies of the virus that are produced when that cell bursts is around 1,000 copies in 10 hours. Now, these are figures taken from in vitro studies done with tissue samples. It's not done in live people, but it's pretty accurate. So the actual period of time from you inhaling a virus to you starting to produce large amounts of virus that start affecting your body is less than half a day. And the time from inhaling the virus to you being infected is less than 10 minutes. Now, SARS is unusual in that it's quite a large virus. This is the size of HIV, Zika virus, flu virus, Ebola, and SARS. Now, it's larger than HIV or Ebola, which normally require blood-to-blood contact to be passed from one person to another. And it's an airborne virus. It's exhaled from your breath, and it's not exhaled primarily in droplets. It's exhaled as an aerosol. That means that surgical masks only limit spread and don't prevent transmission from one person to another, because the size of the pores in a surgical mask are far larger than the size of the virus. Just to put that in context, this is just showing you the size of different materials that are in the air, from heavy dust particles, cement dust, fly ash, etc., at the larger end, to gases at the lower end, and viruses come at the 0.01 level in terms of microns. And SARS-CoV is at that level, where I put the big arrow, and the dotted line is the level at which most conventional face masks will prevent you breathing something in or breathing it out. Okay, back to what it is and where it's come from. It's a zoonosis. So what's that? A zoonosis is a disease you get from an animal host, but it can be contracted and spread in humans. You can pass it from one person to another. You can also pass it to another type of animal, and it can mutate and pass back in a more or less severe form. SARS-CoV-2 passes from other animals to humans, and humans to other species and back. It probably originates in Southeast Asian bats, where most coronaviruses are resident and don't cause the bats any harm, possibly reaching humans through an intermediate species such as the pangolin, if it's come through a benign route rather than some of the alternatives like being looked from a biological laboratory. We'll go into that briefly. Now, this schematic shows multiple reported SARS-CoV-2 mutations that have shown human to animal transmission. Since the start of the SARS-CoV-2 epidemic, we managed to pass it on to gorillas, to domestic dogs and cats, to lions, to mink, ferrets, mice, and hamsters. Probably to lots of other animals, but those are the ones that we've so far identified. And it's been pretty dangerous to certain animals like lions and tigers, which have died from it. Cats and dogs contract it, but seem to be able to cope with the virus, but they can pass it back to humans. And in mink and ferrets, it mutates quite rapidly, and there have been concerns about possible more severe strains coming from mink farms. That was the reason for destruction of mink farms in Denmark and Holland, where new mutated versions of the virus that were thought to be more contagious were identified. Where did it come from historically? October 18th to 27th, 2019, the World Military Games were held in China. A number of American troops were shipped back home because they developed unusual COVID-like symptoms, but were never tested for it because that was before it was on the radar. The first patient that we're aware of was identified November 17th, a Chinese 55-year-old man in Hubei who developed severe respiratory symptoms, thought to be COVID, but wasn't sequenced for it. The patient 0, confirmed to have this new type of pneumonia coronavirus, was found in December the 1st, 2019. And then a number of cases were identified, most of whom had been tracked to going through a wet market where Chinese people buy recently slaughtered animals and fish for domestic consumption. And then 27 cases were identified at the end of December. And this is the market that was on lots of media pictures at the time. The original suggestion was that the virus was spread from contaminated wild meat sold at the Hunan wet market in Wuhan, possibly bat or pangolin, which were both on sale there. The market was closed down and disinfected, and that was thought to have been the original source, and that was the Chinese government line at the time. Another proposal was that it was caused by a leak from a biological research laboratory. Now, the Wuhan Institute for Disease Control and Prevention has a BSL-4 level laboratory less than 200 meters from the Hunan wet market. Biosafety level 4 is the highest level of biosafety precautions used when working with agents that could easily be aerosol transmitted, can cause severe to fatal disease in humans, and for which there aren't any available vaccines or treatments. The Wuhan lab isn't the building below, frequently seen in the media when the WHO team was sent to investigate the origins of the pandemic. The Institute's BSL-4 laboratory is a new facility that's separate from that building and sits behind it. Now, that's the distance from the Hunan seafood market across the second ring road, a couple of blocks down, and in the middle of a highly populated area is the Wuhan Center for Disease Control and Prevention. Now, the BSL lab opened in 2018. It's that grey building on the left with the tower next to it with the glass on it. It's a purpose-built facility for research with high-risk viruses. It was constructed by French engineers, and the staff were trained by French lab technicians. It's the first such facility in mainland China outside of Beijing. And part of the role of the laboratory is to research novel zoonotic viruses, such as SARS, MERS, and SARS-CoV-2. So that laboratory, which was very close to the wet market, is involved in doing highly sensitive work with zoonotic viruses of the type that was the start of the epidemic. But we will probably never know what the true source of the pandemic was. But now the virus is endemic, it's all over the world, it's less important to know where it started. It might have been for containment, but it's not now, it's everywhere. It is important to know that people can pass on the infection to others for many days before they're aware that they're carrying it. Some people are super spreaders, giving off high levels of virus, much higher levels of virus than other people. To understand how it's spread so widely, we need to move from the biology of the virus to looking at human movement patterns. National and international air travel. The biggest factor contributing to the pandemic spread of SARS-CoV-2 in early 2020 was the volume and pace of both internal and international air travel, and the slow pace at which this was restricted. International flights had spread the virus worldwide before any testing was available and before quarantine was introduced. The countries worst hit in the early waves were those with the most heavily used flight paths to and from mainland China. So initially, it was areas like the west coast of the US, the various flight hubs going from China across to Western Europe, and other parts of the world which had less frequent air travel with China were far less affected in the first 1 and 2 waves. Now, quarantine has been used to restrict the spread of viruses and other diseases for a long time. First quarantine law was introduced in Ragusa on July the 27th, 1377, during the bubonic plague. It stipulated those who come from plague-infested areas shall not enter Ragusa or its district unless they spend a month on the islet of Murkan or in the town of Kaptat for the purpose of disinfection. Doctors at the time observed that the spread of the Black Death could be slowed or contained by isolating individuals who might be infected. And the Great Council of Venice had also introduced isolation of travellers from plague areas for 30 days. That was called a Trentine in 1377. And subsequently, they extended it to a quarantine or a 40-day period of isolation. A smaller scale but comparable issue in microcosm to how we prevent international air travel was COVID infection aboard cruise liners. Now, the best known and best studied example was the Diamond Princess. Now, interestingly, cruise ships have soared in size in recent years. Now, the Diamond Princess, I put as a green star on this map, but you can see in 1990, the average size of a cruise liner was about 80,000 tons, and now they're about quadruple that, the more recently launched ships. When we look at the ships that had outbreaks of COVID-19, a large number of cruise ships were basically, held, offshore in various parts of the world because of coronavirus outbreaks. We find that the average size of the average capacity of the ships was over 1,200, crew and passengers, and the Diamond Princess was about halfway up that line with 3,000 or so people on board. Now, this is the Diamond Princess. Now, it was in the media after it was involved in a cruise starting and finishing in Tokyo, and stopping off in Hong Kong and then Hanoi, and working its way back. Now, the interesting thing with the Diamond Princess was that the passenger areas had recently had their air ventilation system replaced with a much more sophisticated one that was cooler and more efficient. The same wasn't true of the crew areas, which were in the bottom 3 floors of the bedded part of the ship. Now, the earlier ventilation system didn't produce much of a rate of COVID. About 4% of the crew developed COVID symptoms during the time that the media coverage was going on. There was a higher rate as they were kept on board the ship for a long time. But the rate of COVID in passengers was over 70%. The rate in the crew was about 4%. The crew and the passengers didn't mix, and their air systems didn't mix either. And this is just showing you the layout, and the blue and green dots are where people were affected on the ship. It wasn't that there was an area that was particularly badly affected. It was that the area on the left side of the picture here had a common air ventilation system. Now, that suggests airborne transmission, and that was right at the beginning of the pandemic being restricted. It doesn't suggest that it's transmitted by sneezing in the vicinity of other people, because most of the passengers were restricted to their cabins, and the only common factor when most of the cases were affected was that they were breathing in the same air that was being circulated around the ship. Having effective test and trace is one way of identifying how COVID spreads through a community. Hong Kong has a very well developed test and trace system, and I'm just going to flash through some slides quickly, showing you how their public health services were able to trace the rapid spread of COVID across Hong Kong through its dance clubs and restaurants. Now, this is from the South Seas Post, I think it was called. I don't think it exists anymore because of the takeover of Hong Kong. But what they do, or did do, were very good infographics that showed you what was happening. Now, these blue squares are the various nightclubs, dance clubs and restaurants that were affected. So the first case was at point 1 here, and they were able to track people from that starting point to 3 others on Hong Kong. From there to Kowloon with a separate place, and then from that particular source through various others. And there were very few cases where there wasn't a line taking you from one particular venue to the next. It was only several days on, when these 2 sites cropped up, where there was no direct link between one and the other. Now, this is a different way of plotting the same information. It's showing you the various venues as you go down the page in chronological sequence. The larger the dots, the older the person. Black dots are male, purple dots are female, and the stronger the lines, the stronger the association between one setting and another. Now you can see that there are dance clubs frequented by people well over 75 in most of these slides. But what it did enable them to do was to track very easily the fact that information or infected individuals went from one setting to another, and that several of these people were what are called super spreaders. They were passing virus on to numerous people with limited amounts of contact. Now, COVID has been particularly severe in large cities, suffering from several factors. The first is high-rise buildings with shared ventilation systems, a bit like the Diamond Princess. The second, large high-capacity arenas for spectator sports. Now, that's true in various venues like New York and London and Liverpool. Large capacity social venues, places like nightclubs, bars and restaurants, and underground mass transit systems with positive pressure ventilation. People will remember I think it was The 7-Year Itch, the film with Marilyn Monroe, where she stands on a grating in a New York street to get the warm air coming up from the underground system in the wintertime. Now, the underground mass transit system in New York passes air from the subway around the streets of New York all the time. And the ventilation systems in high-rise blocks in New York blow common air from throughout the building from massive fans that are in the basement. What that means is that if one person on one of the lower floors is carrying COVID and exhaling it, it will be passed around all of the other flats in the building in very short shift. The ventilation systems didn't until recently have appropriate filtration systems or ultraviolet to kill virus. So the virus was quite happily moved around in an environment that it's quite happy to replicate in. A major issue through the first wave of the pandemic has been the supply of adequate personal protective equipment. Most masks provide limited protection for the wearer against inhalation of airborne SARS-CoV-2, and few masks protect others against exhaled virus from the infected individual. In high-risk settings, staff are now using PPE that would only have been seen in BL4 laboratories, like the one in Beijing, the one in China that I mentioned a minute ago. This is recommendations for California nursing staff now. PPE for COVID-19 must include at minimum N95 respirators or higher, isolation gowns, eye protection and gloves. Surgical and non-respirator face masks don't protect persons from airborne infectious diseases and cannot be relied upon for novel pathogens like COVID-19. A powered air purifying respirator with high efficiency particulate air filters must be worn during aerosol generating procedures on suspected or confirmed COVID-19 cases. So basically, you're wearing a hazmat suit with an isolated air supply for doing any work with somebody who is potentially infectious. Is more effective PPE available for more general use? Just a quote from Aristotle, and yet the true creator is necessity, which is the mother of invention. Since the beginning of the pandemic, several innovative devices have become available that provide greater protection against SARS-CoV-2. Most masks available for personal use can help to reduce inhalation of droplets from infected people who cough and sneeze. Unfortunately, people who contracted COVID-19 are infectious for a number of days before showing any physical symptoms. Some of the work from Wuhan found this pre-symptomatic stage typically lasted for up to 5 days, but in some cases could last for as long as 3 weeks. The virus is passed on in many cases before people are aware that they have it. So, is there equipment that's available for the general public that's more protective. Now, here's one. The UltraMask Mask X is a fan-assisted face mask with a 2-speed fan and replaceable 4-layer HEPA and ultrafine activated carbon filters. The filters last up to a month with 99.97% efficiency at filtering particles down to PM 0.3. Now, because of the Brownian motion patterns of sub 0.3 micron materials like SARS-CoV-2 viruses, this is an efficient system, and the fan makes it more comfortable to wear than a surgical mask. So it's A, significantly better at filtering out viral particles, and B, easier to wear. Now, one thing that's not quite here yet, but I'm going to mention because it's imminent, is the use of graphene face masks. Graphene is an allotrope of carbon consisting of a single layer of atoms arranged in a 2-dimensional honeycomb lattice nanostructure. A charged graphene layer will attract SARS-CoV-2 and kills the virus on contact. A Dutch company had aimed to produce a mask with a charged graphene layer in 2020, but didn't get enough financing. An American company successfully crowdsourced funding, and a graphene mask called the Guardian G-Volt will soon be commercially available. Now, the big advantage of this over a mask that helps you to avoid breathing in the virus is that it will protect both the user and others from SARS-CoV-2, whichever is affected, because it's going to kill virus that you exhale if you happen to be infected, and it's going to prevent you inhaling virus because it'll kill virus that's in the air around you on contact. Now, that's a picture of the Guardian G-Volt mask. If you search for it on the web, you'll find a review of the mask by an American magazine telling you about the fact that it's about to come out, but it's not available just yet. In ASD, difficulties coping with change coping with medical procedures and with unfamiliar clothing are common. There are recommendations for procedures like blood sampling and EEG in the appendices to one of my earlier books. There are some studies on helping with issues such as mask wearing using a telehealth approach, given that often clinicians aren't allowed to have direct face-to-face contact with people with COVID-19 during the current restrictions. And telehealth advice giving and working with families to encourage PPE use by people with ASD seems to be working. Ultraviolet light has been used for some years in disinfecting hospitals, operating theatres. Recently, robotic systems have been developed to disinfect classrooms. They're used extensively in Denmark. Now, that's a robot that can be programmed to go around a school, disinfecting each room, taking about 15 minutes per room, and has to be done when there aren't any students or staff there because of the level of UV that's used, but it completely kills using ultraviolet light, any virus that's in the vicinity. Recently, a system has been developed by Honeywell that's doing exactly the same thing for disinfecting passenger areas on commercial aircraft. I'm going to mention this because it's in the media right as we speak. As we evolve the means to contain and treat SARS-CoV-2, the virus itself continually develops and mutates. Rapid response to recognized issues is critical. Yesterday, the WHO issued a statement on a new variant of concern. CERI, the Centre for Epidemic Response and Innovation in Stellenbosch in South Africa, sequenced a novel variant of SARS-CoV-2 on the 14th of November. The variant had a large number of novel mutations, many affecting the spike protein, which is the primary target of most current vaccines. Since then, over 113 new sequenced cases have been reported. So far, this new variant's mainly been identified in samples sequenced from the Gauteng province of South Africa. This area includes Johannesburg, Pretoria, and Soweto, where reported COVID-19 cases are surging. Few of the identified COVID cases there are sequenced, though, so we don't know whether the increase in cases is an increase in cases of this new variant, although it appears to be. Now, when I wrote this slide 2 days ago, it had been found in South Africa and Hong Kong. It's now been identified in South Africa, Botswana, Hong Kong, Israel, Egypt, Belgium, Germany, Holland, the UK, and the Czech Republic. And travel restrictions are being imposed. There's a presentation by our Prime Minister at 5 o'clock, and I'm sure he's going to be imposing further restrictions in the UK. At present, we don't know what the effects of this new variant will be. The extent of the viral changes suggests it might be able to evade our current vaccines. We don't know whether it's more transmissible, how long it takes from infection to being infectious, or showing symptoms, and we don't know if it causes a similar more or a less aggressive disease. The effects to date have been primarily socioeconomic, government restrictions on international travel, plunging stock markets, and social restrictions. We can't afford to wait and see. If it is more serious and it's not affected by vaccine, waiting for the evidence would be like opening Pandora's box. The sort of measures that I've run through very briefly, using things like ultraviolet to kill virus, using improved masks to minimize the likelihood of inhalation, and going back to doing better social distancing, more people working from home, are the measures that are going to have most of an effect at the moment. It's going to take several months for an adapted vaccine, assuming they can get one that would work with this variant, if the variant avoids the vaccines we have. Okay, how does the current COVID-19 pandemic compare to previous pandemics? Protests are raging around the world. And is COVID-19 an overhyped flu-like issue that we just need to accept and live with? We're seeing reactions across Europe with more restricted government lockdowns and compulsory vaccinations, as we're seeing in Austria, or as we've seen in Brazil, Chile, and saw in the Trump-era USA, an opposite reaction with increasing public demands for access to vaccines and PPE, but with lukewarm government support. What does the evidence show about the extent of the current pandemic compared to those in the past? In living memory, HIV is the only pandemic to have killed more people, between 35 and 45 million deaths in total, but that's over a 40-year period. Mortality from HIV peaked at around 1.95 million deaths per year, and it's now dropped to about 680,000 deaths a year. It's still a significant killer worldwide. Current figures show global COVID-19 deaths have been around 2.4 million per year since November 2020. They're continuing to rise, particularly across Central Europe and the Eastern USA. Now, the best source of comparative data are the Johns Hopkins Coronavirus Research Center and the WHO COVID dashboard. So just to show you what those look like, that's from this morning, a total of 5.18 million deaths so far from COVID, and 260.5 million current cases. This is just showing you what we get from the World Health Organization site. You can get very similar information on both, to be honest, but you can track, how the virus evolves over time, and you can do it by specific country quite easily on the WHO board. Now, this is the history of pandemics, going from the Antonine Plague at 165 to 180 AD, which killed about 5 million people, through to COVID, through 2019 to the present. Now, the balls are different sizes because it's supposed to be in perspective, so just to put them in the right sort of ordering, the Black Death killed 200 million, smallpox 56 million, the Spanish flu at the beginning of last century killed 40 to 50 million, the plague of Justinian killed 30 to 50 million, HIV, as I said, 25 to 35 million, the third plague in 1855 killed 12 million, and then we've got COVID, which currently is sitting at 5.2, but that's going to expand a lot. With the best will in the world, even if we discovered a very good way of containing COVID, a lot of people are going to lose their lives as a consequence of where we are now before it abates. We're dealing with one of the world's most deadly conditions, and it's still here and it's still going strong. The traditional approaches, which Mr. Boris Johnson was recommending at the beginning of the pandemic, Things like using a handkerchief, bend your head forward when you cough, sneezing into your elbow were very common around about the time of the 1819 influenza epidemic, and are still to be recommended, but aren't going to make the thing go away. Just to show you the difference in what happened with the 3 coronaviruses that we've had. SARS, which was 2002 to 2003, killed a total of 770 people before it petered out. It only affected people in 1 or 2 places outside of China. It was in Canada and it was in the US, but it didn't really get a major foothold elsewhere. MERS was a condition that's very similar, but it was thought to have been transmitted through camels as an intermediary host and was first identified in Saudi Arabia. The major concern about MERS was the Hajj, because if MERS had been viable outside of warm climates, then the effect of the Hajj would have been much the same as the effect of demobbing soldiers at the end of the First World War that led to the influenza outbreak then, or the rapid travel all over the place that we've got with coronavirus at the moment. And as you can see, that relatively speaking, there is absolutely no comparison in terms of the numbers of people killed by the previous 2 coronavirus outbreaks compared to COVID-19. They're all relatively long viruses formed of about 30,000 amino acid bases. Each viral copy is sufficiently complex to make exon or exonuclease. Exon is an enzyme that can effectively proofread the viral genome and self-correct errors. Now, that's one of the reasons that it's quite robust. It doesn't get affected by defects that crop up routinely due to environmental radiation and things like that. And smaller viruses like HIV, Zika, and Ebola virus aren't able to correct themselves. So often they peter out because of a gene defect that's incorporated into the RNA. How does risk from COVID-19 compare to risk from other viral infections? This is the top 50 viruses in terms of their risk as ranked on a number of different variables. And you can see SARS-CoV-2 is the second most risky virus after Lassa virus, which is geographically restricted. It's now ranking higher than Ebola.
Dear Ken, I'm so sorry to interrupt you, but your time is nearly there. I think it's already, but we can give you a few more minutes if that's, I don't know how much more you have.
Yeah, that would be handy to just go through a bit more, because I think it would be sad to leave it here.
Okay, sure.
Okay, right. So it's just to show you that, you know, SARS-CoV-2 and SARS-CoV are amongst the most, high-risk viruses in the world. 21 of the 50 highest risk viruses are coronaviruses, and the 12 highest risk viruses are all zoonoses. They're all ones that we get from other creatures. We get lots of outbreaks all the time. There have been 67 separate outbreak notifications in the period since January 2020, when COVID started rearing its head. I'm going to skip some of these slides. Over the last year, there have been between 50 and 100,000 deaths a week from COVID every week. The overall lethality rate's gone down from 1 in 13 to 1 in 49, but that's been accounted for by the huge increase in the number of new cases, so the death rates have continued to rise. Survival's improved, but increasing survivals caused growing numbers of people to have long COVID. We've now got a WHO definition of long COVID, and between 10 and 30% of all adults who survive develop what's called long COVID. They have problems with a whole range of physical systems of their body. The issues depend partly on physical health and infection and the primary mechanism by which they're infected. And the effects compromise a whole range of different things. I'll skip these because they're in the reading list. In children, we get what's called MIS-C, multi-system inflammatory syndrome in children. And MIS-C is a COVID-related problem where you have ongoing problems which are outlined on this slide, which you'll get in the reading list. But a large number of assessments are now available for COVID-related issues in Arabic, Bengali, Hindi, Iranian, English, Hebrew, Italian, Turkish, Urdu, Vietnamese, Russian, Spanish, Serbian as well. I've got the full list of languages on the reading list, together with the function and what the assessments are supposed to do. These are the scales that are available at the moment, which give you a profile of people. And these are long COVID support websites. Now, we're not going to know the extent to which people with ASD are at greater risk until we've got data which we don't have yet that's been properly analysed. When we look at the SARS epidemic, it was in 2003 to 4. There was a link with Kawasaki disease and SARS, and that was linked to autism, but only in a very small paper that was published in the American Journal of Neurology. A more recent paper in 2011 looked back at 750,000 people and found a strong association between Kawasaki disease caused by SARS and subsequent diagnosis of ASD. What they couldn't tell us was whether the ASD came after or whether the ASD was a risk factor for Kawasaki disease. In the early waves, various reports from the UK and Italy noted greater numbers of children and adolescents with ASD being admitted with COVID-19. These were mainly affected by the earlier variants. They tended to be admitted for longer periods than other children, tended to recover. More were from other ethnic backgrounds than Caucasian backgrounds. They tended to have a higher body mass index than others, and a greater proportion had pre-existing cardiovascular issues. It may be that the body weight cardiovascular issues were the risk factor. Numbers are too small to calculate so far, but the references to all that works in the reading list. Now, we can look at physical areas that are affected by COVID-19. People can look at these at their leisure. Interestingly, when we look at the various areas, we find that the kidney uses a different thing to ACE2. ACE2 is the receptor for SARS virus. And you don't get ACE2 in kidney tubules. What you get is stuff called colectrin. And in the kidneys, you don't find evidence of viral RNA, which you do in virtually all other body tissues. So we're starting to gradually piece together some of the neurobiology. The other thing is that loss of sense of smell is very common in ASD. And that's because in some cases, the virus infects the body through the top of the nasal cavity directly into the olfactory bulb at the base of the brain. And that's why you get these areas of the brain showing up evidence of virus in various types of nerve cells, oligodendrocytes, astrocytes, microglia, and neurons. And it spreads through the blood-brain barrier into your bloodstream. And we know, very last 4 slides and I'm done. This is looking at genes in autism, schizophrenia, mental, major depression, etc., and links to what you can culture from cells from lung tissue, and whether you get up or down regulation of specific cell groups. And what you find is that you get up regulation of 3 particular genes that are strongly linked to autism in the literature: 10, FKBP5, and SYN2, and you get down regulation of several others: MTHFR, AKT1, and EHMT1. We're starting to see a profile where people with autism show unusual patterns of reaction to COVID. They're less likely to die from it, as far as we can tell, but they're also, we're seeing a genetic profile which is linked to particular patterns of reaction. Okay, I'll leave it at that, and thank you very much for letting me run over time.
Thank you, Ken. Thank you for this presentation. I guess there is so much unknown regarding this COVID-19 and how it's actually, how it's affecting children, and not to mention the children from the spectrum. So yeah. I'm sure that in time to come, we'll hear more of the new research, and I'm sure maybe in years to come, we might find out more. So
there is a huge amount of research. I've given you a 20-page reading list to go through. That's just just references. But there is so much that's being done, but a lot of it is on very small groups, and a lot of it is on earlier phase versions of the virus. By and large, children aren't affected by COVID and don't get admitted to hospital. Children with autism are more likely to be admitted. They're more likely to be in intensive care for longer, but they're less likely to die. They have a different reaction to the virus. And in some ways that's positive. What we don't know is in terms of long COVID, are they left with lifelong problems as a consequence of recovery?
Yeah, what are the consequences, right? Yeah.
And that's what we really need to be looking at. I mean, I'm now leading the clinical service for long COVID for the Liverpool area in England. And we get about 80 referrals a week, something like that. And it's a new service. We're not quite certain what we need to offer, but with things like sleep problems, anxiety, difficulty coping with change, and all the changes in containment issues, etc., that are happening all the time because of changes in what's happening with COVID, must be incredibly difficult for many families to cope with.
Right, sure. Yeah. Very interesting. Thank you so much, Ken.